So let's get started. Welcome back to the second slot. So that was the initial lecture outline. We have covered part 1, ecohydraulics and life in moving fluids. That was designed to provide the background to address the second slot, which I've titled Fish Behavior in Response to Hydrodynamic Stimuli. Again, this second slot is divided in 2 lectures. In the first one, we're going to talk about brown trout and how brown trout chooses space or habitat based on the hydrodynamic properties of the habitat itself. In lecture number 2, we will talk about eels downstream migration. So let's start with the first. Assessing hydrodynamic space use of brown trout. Before I start, some acknowledgements. All the material that I will be presenting is essentially taken from this paper that was published in Journal of Experimental Biology in 2016. I want to acknowledge the contribution of Jim Kerr, my PhD student, and Paul Kemp, the other supervisor, my colleague in Southampton when I was based there. The title of the paper aligns with the title of the talk. Nothing strange. What we wanted to do here in this study was getting a little bit a better understanding on how, not much about how fish swims while migrating, but actually how fish, when not migrating, chooses his space, his habitat, as a function of only hydrodynamic properties of this habitat. Okay, we wanted to isolate the effect of hydrodynamics on space use. That's what it's called. And this clearly has lots of important implications. Understanding how physical environment influences the distribution and movement of fish is a fundamental theme in ecohydraulics. We need to understand what are suitable habitats for fish under, as a function of many variables, food availability, heterogeneity, and blah, 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 everything that describes the ecology of the system. But what's the role of hydrodynamics? What is really the driving principles that moves fish towards one place or another? How do they choose where to stay and where to hold station, or where they spend their time, essentially, in the aquatic environment? This topic has created a fairly big debate in the literature, because if you want to associate what hydrodynamics, if you want to identify what sort of hydrodynamics fish prefers, you have to find an hydrodynamic matrix to describe it. Is it fish, do they like low velocity areas to stay there? Do they like high turbulence intensity? Do they like certain values of the skewness, of the flatness, of velocity, of any statistics that you can think of? And there is a huge number you can use. And choosing the appropriate hydrodynamic matrix to describe the habitat and the suitability of the habitat, again, has been the subject of controversy. And the reason is that traditional methods, essentially, when I say that there is a controversy, I'm saying that there are conflicting results in the literature. For example, some people say fish like to stay in highly turbulent waters. Some others say no, they like to stay in calm waters. Why? Are they looking for food? Are they resting? What's going on? And one problem when those studies that try to isolate the effect of hydrodynamics on the space use tend to heavily rely on simple correlation between a single point, time averaged velocity statistics, and space used by fish. So traditionally what you do experiments in a flume, you put some sort of rocks or sources of complexity for the hydrodynamics, then you put the fish inside the flume and you monitor with the camera where the fish goes. You see what sort of preference they have for different places, and you correlate where the fish stays longer with an hydrodynamic metrics of that place. And what they do is they use time average single point velocity statistics. However, as we have learned in the previous slides, a single point, so the characterization of the velocity statistic at one point does not capture, fails to capture flow-fish interaction where flow properties varies in time and space, simply because it's a one-point description. So they would fail to capture Karman gating, for example, where you have movement of fish, slalom zigzagging among vortices. That would be very difficult to capture with one-point measure. Or entrainment, where it's a whole flow field that really describes why the fish decide to entrain around the cylinder. So that's a shortcoming of traditional methods. The other problem is that attempt to quantify hydrodynamic space use by fish was addressed in field experiments, and these obviously produced mixed results. And again, this is not surprising because in the field experiments are extremely complicated. There are many confounding variables that are impossible to control, like food, presence of predators, competitors, mating issues, and all these variables will hide the real effects, will make impossible to isolate the effects of hydrodynamics. The third reason is that previous papers tended to, again, find statistical links between space use and turbulent flow characteristics, velocity statistics, that have no clear biological meaning. So some people say, okay, I've noticed that there is a strong correlation between space use and the turbulent shear stress. Why? Why would the fish stay in a region of high shear? What's the reason for that? What's the biomechanics behind that? So it was sort of randomly selected. It was just an observation without a clear biological link. So I decided to take a very easy approach, in my opinion. We made a couple of hypotheses, which are fairly intuitive to me. Hydrodynamic space use is dictated by energy conservation. So essentially, this is a general principle. If fish can save energy, so has energy then to do other things that are important for his life, finding prey or escape from predators, mating, and so forth. And more specifically, the the hydrodynamic space use choices are dictated by strategies that minimize the cost of swimming, that, as we have seen, have two sources, thrust and drag, which are really, really difficult to quantify for a fish that swims in a turbulent flows in a complex environment. But anyway, this is the first hypothesis we did. The second hypothesis was that in an hydrodynamically complex environment, we can explain space use by means of two things, essentially. One is the specialized behaviors that we have observed earlier on, so entrainment, bow, wake riding, Karman gating. These are all strategies or specialized behaviors that allow to save energy. And then we have built an ad hoc drag function that, whenever these specialized behaviors are not feasible, are not possible, allows to quantify energy expenditure, essentially. Okay. This drag function D, there's nothing new. Perhaps it's a bit new in the world of fish, but definitely not in fluid dynamics. But the way we have built it goes as following, like following a simplified example. We say let's consider just an object of a standard shape, like a cylinder or a disc in a 2D problem. In the absence of turbulence, you put an object in a current. The drag exerted on the object is proportional to the square of the velocity. That's what we say. We say fish, the drag on fish depends non-linearly on velocity. Okay, we are in the inertial regime. We are far away from the low Reynolds number. So that was the first hypothesis. And the other hypothesis is if we add turbulence, as like a fluctuating vertical or lateral component in this case, the drag increases. Okay, without turbulence would be related to the square of the velocity. Whenever you add, in an idealized case, a fluctuating lateral component, the drag, if you do the math, becomes proportional to the velocity multiplied by the square root of the sum of the squares of the two velocity components, u and v. This is very easy to show, and this is due to the fact of the nonlinearity dependence between drag and velocity. The reason why we wanted to put some turbulence in this ad hoc function is that turbulence, in our opinion, destabilize fish. I mean, there is quite a bit evidence on that, despite the results by Waldnikor that we have seen. But clearly, it must have, it should have an effect. If not, well, the idea is to test this hypothesis with experiments, but we thought this was the best candidate to describe energy consumption by fish while swimming in a hydrodynamically complex environment. So eventually, we decided to, let me comment a little bit more this drag function. Essentially, what this drag function says is that imagine that you are riding your bike. Okay, there is no wind. You're riding your bike at a velocity u. You receive, you feel a drag force that is proportional to u squared. If you have a lateral component, so some wind, for example, starts blowing from a side, it's going to be more tiring for you. You probably have experienced that. And that's because of the nonlinearity between drag and u. That's what it says. If you imagine that these velocities change rapidly, you can define this drag function as u multiplied by the root mean square of the sum of the square of the velocities, and you sort of capture that. Clearly, in a turbulent field, you don't have a constant lateral velocity or even a constant velocity changing sign. You have, that we decided to take as characteristic velocity of turbulence, the standard deviation of velocities at each point, okay? Clearly, in experiments, we are in a 3D condition, so we have to consider the lateral and the vertical component. To test this hypothesis, we decided to do some flume experiments at the University of Southampton. There is this large flume, which is well suited to do some fish research. The flume is long 18 meters, I think, and roughly 1.4 meters, not exactly 1.5, but fairly large. We have selected a test section of the flume, roughly 3 meters long, which was bounded by a, upstream by a honeycomb sort of a flow straightening device that we wanted to have to minimize any fictitious turbulence coming from upstream, which is unfortunately in that flume is fairly high. Then we play, in order to create some sort of hydrodynamic complexity, we have placed some cylinders of varying diameter from 9 centimeters down to 1 centimeter, which were placed in two different arrangements, increasing and with this arrangement identified by treatment as treatment B in this figure. The idea was to create an hydrodynamic complex environment with some sort of specific eddy size generated in the wake of these cylinders. On the downstream part of the test section, we put the mesh screen with a release chamber for fish, meaning that before each experiment, we would put the fish in here, wait for a while so that the fish would climatize, and then we would release a gate, open a gate in here so that the fish could start exploring this hydrodynamically complex environment and choose positions he liked, essentially, so that we could correlate then the hydrodynamic properties of that place with the choice of the fish, essentially. Besides treatment A with the cylinders in this configuration, treatment B with this other configuration, we also did what the biologists call a control experiment. So the same experiment, exactly the same thing without cylinders. Okay, so very, very, very simple, and we repeated that. And I think that's it. What sort of fish did we use? Brown trout, again. The source, we got some hatchery fish, and we have divided it in a small, medium, and large size class, ranging from with a length of 14 centimeters up to 28 centimeters for the large trout case. Quite a few number of experiments. This is very time-consuming and very difficult experiments to do. 53 trouts for the small, 25 for medium, let's say, and 26 for the large trout. We also catched some wild trouts in the field, which were mixed in size, were like 19 centimeters plus minus 3.6, essentially. We wanted to have both hatchery trout and wild trout to see if the results were actually, if the hatchery results were aligning with wild trout results, so that any result we got was just a result of being grown in a hatchery, okay? Because what we are interested in are actually wild fish. But obviously there are regulations. You cannot fish many, and they're difficult to fish. It takes time. They die when you transport them to the lab. I mean, it's you need to have the proper facilities to maintain them in a hydraulics lab. It's a lot of work. All the experiments were done by placing a camera on top of the fish, putting some infrared illumination. All the experiments were done at night, so zero visibility. The fish would choose their position just by sensing the flow by means of the mechanosensory system, the lateral line. We didn't want to have any side effects, essentially. So that we could monitor its position with also not exactly automatically, but as automatically as we could. We did careful measurements of the flow field with an ADV, which is not, let's say, particularly great for turbulence research, but it works fine for this type of studies. ADV allows to make one-point measurements by sampling time series of velocities in all the three components of the velocity vector. Frequencies vary between 50 and 100 hertz, but that doesn't really matter because the sampling volume is fairly large, so the measurements are filtered. But I mean, ADV are okay to measure the bulk properties of the flow, like up to the second order statistic. I think we are fine. So we could plot the flow field for the mean velocity, the turbulence intensity, which is essentially the variance of the velocities, the relative turbulence intensities for any component, the turbulent kinetic energy, the shear stress, the Reynolds shear stress, which is the covariance between the longitudinal and the lateral velocity. Please keep in mind that in this configuration, the wake of the cylinders would have just one vorticity component, so would be eddies mainly with one direction. All right, it's a fairly 2D problem, which allowed us to monitor the flow field only at one plane and to make things relatively easy. We have chosen to address these turbulence metrics because these are those that are under investigation or were under investigation, were investigated in the past literature. So saying the turbulent shear stress, this is the eddy size that we have estimating actually using 2-point correlations. We didn't trust the Taylor hypothesis within the near wake zone, so we could actually estimate the size of the eddies by means a little bit more carefully. And as you can see, it's a fairly rich in hydrodynamics domain, you have different eddy sizes, you have regions with high velocities, low velocities, high turbulence intensity, so high velocity fluctuations. And of course, we have mapped also our ad hoc drag function that we have defined. As you can see, is not that different from the in patterns from the mean velocities, but there are some difference, and these will be actually will have also an effect, as we will see, a slight effect on the space used by the trout. In doing this sort of experiments, we had to find a way of quantifying space preference. Okay, we wanted to build up a way to quantify what fish prefers. And this is not that straightforward, because while moving fish, none of the fish moved enough to experience the whole flow field. Okay, so we did decide to define the space used, and this is identified by these blue points that are essentially the snout position in time of the fish. Okay, so that's where the head of the fish is essentially. And then we have identified the sampled, so what actually what the fish experienced while navigating in the test section. And that depends, as we have seen, by the mechanosensory system they have by the lateral line, which according to literature allows to have an idea of the hydrodynamics within this range. So within the field of detection is, say, 2 body lengths long and 1 body length wide around the cylinder, so that we could map the sampled hydrodynamics and the used hydrodynamics. Hydrodynamic preference, how did we define it? Essentially what we did, by monitoring the space used, we decided to take an hydrodynamic matrix. Here I have taken by example the drag function that we like a lot. We say, okay, this is the fish, the space that was used by the fish, and we have reported that in a frequency distribution plot. Then we did exactly the same about how this drag function was common within the space sampled. Hydrodynamic preference was just defined as the ratio between space used and space sampled, just to reflect the fact that we can only define the preference the fish showed within the hydrodynamic environment he managed to sample, to feel. Okay, that was the idea. Furthermore, we identified all those areas within the test section where specialized behavior zones could actually occur. If you remember, we talked about Karman gating, which was known from the literature. So by analyzing the data from the numerous papers on Karman gating, we could get a sort of a definition of an area where Karman gating could occur, which depends on the cylinder diameter and the size of the fish as well. So we could identify it in our test section. The green rectangles identify where fish would go to do entrainment. Again, we could get that from the literature. Bow riding, again, there is fairly robust literature that allowed to find potential areas where fish could do bow riding. We also observed some new sort of strategies. One is tail holding, is pretty trivial. Essentially, some fish were very, very lazy, and they would put their tail against the downstream screen and their head against the lateral wall, and they would stay there without moving. So that's not valid. And that occurred really, there was a lot of clustering of points here and here identified by the black rectangles. The other one, which is less straightforward, we notice a lot of fish staying in this area, And to be perfectly honest, we don't have an explanation why they really liked that position, because that's actually, again, an area of very fast velocities, which would suggest high drag conditions, right? So high energy expenditure locations. However, it's not that far off from areas where the fish wouldn't train, so we have interpreted it as a sort of entrainment. But that why there is energy saving in proximity of a lateral wall and a cylinder, we are not that sure. Okay, but again, these are the potential areas at low energy cost. Okay, for the fish. Again, so just to summarize, we have hydrodynamically complex environments. We could classify with velocity measurements. We had put fish inside, and we observed fish movements within this test section. And we now have a tool to quantify preference expressed by the fish to different locations. And these are the results we got for the drag function now. Here, these curves on the left vertical axis, we have the preference as defined previously, pH, and different lines are associated with trouts having different dimensions, small, medium, large, and the wild class, okay? The histograms, the gray histograms instead are the frequency distribution of the whole available hydrodynamics in the flow domain, okay? Meaning if these curves followed, the preference curves followed these patterns of the histogram, that means that they didn't really like it. They stayed there simply because that's the most probable hydrodynamic metric. Well, in this case is the most probable drag that you can have, so it's more likely that to experience it because it's everywhere, okay? If these curves diverge from this maxima, then there is a preference which is meaningful, essentially. Now let's forget a little bit about figure B. Let's look at figure A. When we did, we computed the preference for small, medium, large, and wild fish using the ad hoc drag function that we defined. We noticed that there was a very clean results of a bimodal distribution. Essentially, there were a strong preference for really low drag areas, definitely lower than the available, the most probable available hydrodynamics, and but also a peak at very high drag. And this was surprising at the beginning, but then we decided to filter out all the points that belonged to the to these SBZ, the specialized behavioral zones. So we took out all the points belonging to these areas, and essentially what you get is that this peak disappears. So this preference in this drag at this high drag are explained by the fact that the fish was at locations where he had to use not well, he was using either Karman gating or entrainment, which is associated with very fast velocities, okay? But yet was experiences low energy consumption because of these specialized behaviors we talked earlier on. So this picture suggests that wherever these SBZ do not occur, fish like to stay in zones at low drag, okay? And to compare now the performance of the drag function, so this result just says we were right, the drag function works, explains nicely the space use, the hydrodynamic space use of brown trout. So what we did, we then filter out all these points of SBZ, neglecting the behavior in these specialized zones, and we compare the performance of the drag function against many other metrics. In this plot, you have the results of treatment A with the cylinders decreasing from left to right, the treatment B where they were were put in another configuration, and the control experiments, essentially an experiment with no cylinders in the test section. On top, here we have the preference curves for small, medium, large, and wild fish, which are the colored curves again, and the metric series drag. And again, there is the gray histogram, which identifies the available hydrodynamic, the available drag in the whole test section. Okay, and you can see there are quite spike and well-defined peaks, defining a clear preference of the brown trout in both treatment, meaning that there seems to be no influence of cylinder configuration, and also in the control case. Preferences are quite high, especially for the small trouts and the wild trouts, which were mixed. In the wild class. There were small and large trouts. The fact that preference is higher for small trouts, it can be explained because small trouts need more to save energy because they are smaller, so they have less power and they are more interested in saving energy than a very large trout that instead can swim almost undisturbed within the experiment. Even in the control experiment, we observe the same results. Interesting, the small trouts, here the blue curve, actually has a peak towards even lower values of drag. And that's because being smaller, they can go perhaps even closer to the lateral walls where there are smaller velocities and so smaller values of drag, something that the larger trout cannot do simply because of his size. Now, this is the shear stress, which was a big candidate according to literature. The preference follows the histogram, so it's not very meaningful in our opinion. And also the preference itself, the value ranges from 1.2 to maybe 9 versus the much higher values of the drag function. And overall, there is not a consistent picture from treatment A, treatment B, and control, actually, for the shear stress. Similar story for the turbulent kinetic energy, which somewhat follows the patterns of the histograms. Not much in the control experiments, but keep in mind that here the variations in turbulent kinetic energy were not that big. As you can see, that the range of turbulent kinetic energy that is dictated by the cylinder experiments is a lot larger than the control. So anyway, this seems to suggest that drag the drag function explains a lot better than the other metrics space use. The question now is how drag compares to the mean velocity. Okay, we may actually overdo that. Again, on top we have the drag function with the preference curves. On bottom, the preference curves obtained from the mean velocity, which is the simplest metrics that is actually used in many models. To be perfectly honest, also the mean velocity works pretty well, although the results are a bit more noisy. Meaning that the drag function seems to perform slightly better, not significantly probably, and the explanation for that is that essentially the sigma, the variance of the lateral and vertical velocity within the test section were anyway smaller than u squared. So the difference between the drag function and u squared or u, it's minimal. Yet there are some drawbacks associated with u that are overcome by the drag function, which seems to give cleaner results. Another question that arises is why fish would choose this particular value of 0.12 for drag. We haven't investigated this much further. We haven't even normalized this value with something. We think it may actually depend on the experiment. I mean, maybe experiment dependent. So if you manage to build another hydrodynamic field, perhaps you would see that allows the trout to explore even lower drag areas. They probably might, but so I wouldn't say that this is a a universal result. It's probably experiment dependent. But clearly, I think these results are enough to say that fish do like when low drag areas. Okay, fish, whenever they are in the hydrodynamics influences fish behavior only on that. There's not much to talk about. So fish like to save energy. Conclusions. We said at the beginning that there is much debate on what is the most appropriate hydrodynamic predictor for space use. Because of the shortcomings we believe in the previous papers. When specialized behaviors are ruled out, like entrainment, Karman gating, and so forth, what we used, the proposed drag function, seems to explain why it seems to be a robust, simple, and biological relevant metrics to explain space use. And the other comment I would like to make is that a similar drag function was used to explain not much space use by migratory routes. So the use of these metrics can be extended to explain also the preferred routes by fish while migrating. An example is the work by McElroy et al. Of 2012, where they have monitored, I think, the migration of sturgeon upstream. Okay, where if you migrate upstream, energy expenditure is very a relevant problem. Think about a fish that has to do thousands of kilometers or hundreds, depending on the species. Any delay, any over energy spent would delay the reaching the spawning habitats, or actually would prevent the fish to reach them. So it's very important to save energy. And they have observed that the sturgeons like to migrate on the sides of the banks where velocities are very low and presumably drag is very low, and therefore energy expenditure. And this makes a lot of sense, as we have discussed. But I will leave you with an open question. So what about downstream migration? Whenever you have downstream migration, you could actually be drifted nicely by the water. So energy expenditure intuitively should be less important, and this is something that we will address for eels after the break of 10 minutes. Okay, thank you. Any question? Anything? Okay, let's get back to fish. Not grilled fish, but we are now on the second part of this slot, and we have finished the previous talk about talking about what about downstream migration? What are the preferential routes? Is there any? We said the drag shouldn't play any role because now fish are drifted downstream essentially. So definitely energetics should be less important. We carried out a study about eels downstream migration. Clearly, all these studies, whatever you find, is species dependent. We chose to study eels because it's an endangered species, and the eel stocks around the world have depleted heavily in the past 30 years, and it's considered now an endangered species officially. All the work that I'm going to present most of it actually, not all of it, but most of it is contained in this paper, which is published in the Proceedings of Royal Society B. And I want to acknowledge the contribution of Adam Piper, postdoc, at that time was a postdoc at Southampton, Fabio Siniscalchi, Andrea Marion, postdoc and a professor at the University of Padua, Rose Wright from the EA, UK, and again Paul Kemp, my colleague, former colleague in Southampton. Let's talk a little bit about eels. Eels are mystery animals. Their life cycle is really incredible to me. When I read about eels, I was really surprised. Do you know about that? Have you ever heard? Yeah, somebody says yes. One, okay. So let me just summarize the life cycle of eels. All the eels in Northern Europe and North America are start their life in the Sargasso Sea, essentially. And so from the eggs, they become leptocephalus. Correct pronunciation? Very good, thank you. And then they are born here, and then they start being drifted by currents, essentially, and going back to rivers and lakes in in Europe and in North America. By the time they arrive in fresh waters, they are glass eels. That's when they start migrating upstream. While they migrate and they live in the fresh water, they become elvers and then yellow eels, so they grow larger and larger until they get this silver color, become what is called silver eel, and then they have instinctively the need to migrate downstream and to go back to the ocean. And what they do is that they travel apparently all the way again to the Sargasso Sea to for mating, essentially, which is the spawning, the supposed spawning area. But nobody has ever proved it, has ever tracked eels from North America or Northern Europe to the Sargasso Sea. There is a very recent paper, I think, 2016, big group of scientists who managed to track few eels that were released in Ireland, in Sweden, in Norway, and in France and Spain as well. And they managed to track them with some fancy techniques, and the trajectories seems to suggest that all these are actually going to the Sargasso Sea, but they lost signal of all these fish while moving. What we are interested in this talk are silver eels. Okay, so what we are going to talk about are big eels, roughly 1 meter long, that are moving downstream in fresh waters. That's what we are aiming to. What I'm aiming to show you today. As I said, there is the severe decline of the European eel because there are, while migrating, both upstream when they are glass eels or downstream when they are silver eels, the migration is delayed or blocked by river infrastructures I was showing, like dams, weirs, and so forth. Eels suffer really high rates of mortality because they are susceptible to impingement at exclusion screens. Whenever you have a water intake or something, the asset owner has to put some sort of screens to prevent the passage or trash racks to prevent the passage of debris and fish itself. And they get impinged, they don't have the power. If the velocity is high, they don't have the strength to swim back, and so they get stuck in these screens. There is general lack of knowledge about eels' behavior in correspondence of man-made structures like dams and weirs. But it seems that eels are what is called thigmoctatic, so that they like to go there, to touch the structure, to assess what's going on, and then take a decision based on that. So they are more prone to impingement, because if they didn't go close to the screen, probably they wouldn't be impinged. Generally, all the downstream passage solutions that are tried worldwide have variable efficiency, effectiveness, but in general is slow. And so this is an emergency. We need to understand how eels actually behave in correspondence of these structures, so that we can design these structures in a way that is friendly for eels. Again, it was the same concept as explained this morning. We need to understand what really attracts eels so that we can guide them towards a safe passage, and what repels eels so that we can find ways to repel them from dangerous areas like water intakes for turbines or screens. This is a very sad picture taken from the literature of an example of impingement in a trash rack of many eels, and you see how many there are. And besides, in a water intake, and besides being very, very sad, this has also a tremendous and high cost for the for the water companies, because these trash rocks have to be cleaned daily, and the screens get clogged, and so in dams they lose head that can convert in energy and so forth. So besides being an ecological emergency, there is also an interest by industry to get rid of these eels because they cost money for them. Okay, so it's a very, very important problem we want to solve. We want to save the eels. To do that, we need to get an understanding of how they behave. In an attempt to do that and to understand what they do, I already telling you I will give no answers today. Essentially, the outcome of this study is we don't know. But we have some new questions, which hopefully are sharpened with respect to previous literature. Okay, I was saying, in an attempt to understand how eels react to hydrodynamic stimuli, we decided to carry out some field experiments in a very, very special place. This is a channel located in southern England. The channel is, okay, the flow direction here is from left to right. There is previously this was a water intake for hydropower generation. Now it was made redundant, but still there is a bar rack in here which is sloped, which can be used to constrict or open the flow. Essentially, you can alter the hydrodynamics as you want using this bar rack. And this is the bathymetry. The color scale is in here, is in water depth. The water depth ranged from 1.8 meters to 0.54. The flow, there's nothing in here. The flow is all diverted in there. We decided to essentially do two experiments. One, when the gates of the bar rack were fully open, which implied very low accelerations. And the other experiment, we call it constricted, where we have constricted the flow to create very strong acceleration in proximity, of the bar rack. What we did essentially was to put some eels in here further upstream from the rake and monitor their movements. I'll show you how, until they escaped. Okay. And we monitor that under the fully open, which is UL treatment, and the constricted conditions. That's what we did. The nice thing is that we could carry out the experiments by keeping the same discharge. So the volume of water per unit second flowing in the river, because there was upstream here, there was a weir that allowed to divert some water when some natural oscillations of the river would put the discharge higher. So we could actually control it very, very nicely. It's an excellent field site because you can control the hydraulics as you want. I was saying before doing the Eels experiments, we did some transects with an instrument called ADCP that allowed us to measure mean velocities in few cross sections of the channel, and these are the black line. And just a little bit of background on an ADCP is a big instrument with a huge sampling volume. Okay, so what that actually measures at when flow depths are this shallow is the bulk, is the mean velocity, is the depth average velocity, nothing else. So nothing really fancy, no turbulence whatsoever. Okay, but we could at least map the flow fairly well. We also put some antennas in here in these red cross sections to monitor essentially at which depth eels were swimming. Usually they swim very close to the bottom. We wanted to verify that, and actually that was the case. We used 20 eels per treatment, so 20 eels for the fully open experiment and 20 eels for the constricted experiments. I forgot to mention that we wanted to create a sort of an area with high and low accelerations because, according to literature, acceleration I'm talking about convective acceleration. Okay, so the flow is steady, so it's the acceleration due to the spatial heterogeneity of the flow. Okay, that's what I'm talking about. I was saying it seems that this acceleration is what other fish species respond to. For example, the work of Goodwin and co-workers show that really salmonids respond to acceleration. I mean, the response is pretty clear. When the acceleration is low, they tend to mill. When it's very high, they get scared. They make some rejections. They go back upstream, actually, or downstream, depending where they are migrating. So our hypothesis was: is acceleration the right metric also that determines some behaviors to eels as well? And the idea was to test this hypothesis by creating low and high acceleration zones here to see the response of eels. I was saying we used a total of 20 eels per treatment. Experiment again were carried out at night, not much needed because visibility in water was very low anyway, but we decided to do all the experiments at night, which is also when eels are very, very active. We used acoustic tagging to monitor eels, to track eels when swimming in the channel. Essentially, what you do, you put a lot of microphones in here. You put a tag under the skin of the eels, and the microphones are able to detect the signal. And by triangulation, I think they work out. You can work out the trajectory. The accuracy is. After a little bit of analysis, we worked out it was roughly half a meter, between half a meter, definitely less than a meter. Okay, not great, but not bad. Keep in mind that, just to give you a scale, these are 10 meters, so the channel is 15-20 meter wide, and they had to swim through a length of what, 30-40 meters in total. Okay. We verified with these antennas, that eels swam at the bottom, which is an important information for what I'm going to tell you very soon. To back up the measurements, the ADCP velocity measurements, we decided to do some very simple, basic numerical simulations of the flow within the channel, so that we could have a much more detailed flow description of the field site. We used the velocity measurements that we did to calibrate this model. The model is we did 2D simulations. So essentially what we are simulating are flow depth and mean velocity with a very rough K epsilon closure scheme. And these are the results we obtained. This is the velocity field, the color bar is in here, and this is the acceleration field, which I've already told you seems to be an important metric to interpret eels' behavior. As you can see, the fully open treatment has a fairly smooth exit from the channel, whereas the constricted treatment has a zone of very, very high velocity in proximity of the barrack, and of course a high convective acceleration. I forgot to put a picture. I mean, the agreement between the measurements and the simulation was not bad at all. We managed to capture fairly well the flow depth and the mean velocities, except for for a few areas here we had some problems, and a little bit here. But overall, very good agreement. I always say you can do very good simulations if you have very good measurements, so that you can tune your model as you want, essentially. But that was the case, and we used the model only to refine the flow description. Now, to interpret the behavior of eels, we needed to define some sort of behaviors. Essentially, what is behavior? You need to quantify it somehow. And first, we looked at the eel's trajectory, and as I will show you, what they did was basically swimming, almost drifting passively in this first part. And then once they reached this area, they started doing some crazy things. They started going back, start milling. So we wanted to interpret the behavior in this area where things got a little bit less smooth for them. So we defined switch points, switching behavior, okay, from a passive drift behavior to something that is much more active. We defined rejection when downstream moving fish switched abruptly from negative to positive Reoth axis, meaning following the current going back, essentially, so it changed direction completely, that's what that means, and moved in a counter streamwise direction for at least 3 meters. You may say, this is a subjective definitions. I agree, but what do you do? It's very difficult. And this is also a definition that is used to describe fish behavior in the literature. So it was our best shot, at least consistent with previous studies. We also defined exploratory behavior when downstream moving fish switched from negative to Reoth axis to some sort of lateral movements, greater than 3 meters length, perpendicular to the streamwise flow, and at least encompassing more than 2 turns. You know, it's it's very arbitrary, but you know, you can tell by eye. You know, you can interpret the behavior of a fish just by looking at the trajectories, but quantify switch points in a rigorous way, non-arbitrary, it's very difficult. So this is the best we could do by matching intuition with quantification. Okay. Sorry. Yeah, it's definitely subjective. It's probably if you give the same data to somebody else, they would see something else. But this is one of the problems. But anyway, in my opinion, one of the, if not the best result of these experiments was not much about the switch points, was how eels swam upstream. And look at these trajectories. 37 over 40 eels actually moved downstream. 3 eels decided to do whatever they wanted. They went upstream. We lost them. Okay. But 37, so a good percentage, went out through the barracks, and all of them swam in these 2 corridors in the channel with a velocity that was about 0.7, 0.6, the depth averaged velocity. Now, except for this eel here, that actually touched the side banks, all of them did not come into contact with the banks. And yet, they decided to go into these 2 corridors. None of them went through the center. Why? Weird. Very, very strange. I was saying the velocity was 0.6 with 0.7, the depth average velocity, which makes sense because they swam at the bottom where velocities are slightly lower and they were drifting by the near wall velocity, okay? So this is the first interesting results that we will discuss later on. Now, 35 out of 37 eels, so all of them showed a reaction when they approached the rack. Okay, now you can tell your definitions of rejection, of exploratory, how did I call it? Exploratory behavior is arbitrary. You can see whatever you want. Fine, but it's definitely an objective result that all of them reacted. All of them stopped being drifted. So they either stopped or got scared. Okay. Interestingly, in the open, fully open experiment, 75% of the eels showed exploratory behavior, okay, not rejection. Whereas in the constricted behavior, all the eels went nuts. So once they approached the barrack, they started swimming upstream and doing really, they were clearly scared, okay. every time there was a rejection, nearly 91% of all the fish they rejected did that multiple times. So they tried to pass, they got scared, they went back, they swam a little bit, then they went back there, they got scared again, then there was some habituation, and eventually they went through the rack. Okay, so that was what happened. Just to give you an idea of what exploratory behavior and rejection looks like, so in the constricted case, the eel moved here, then turned, decided to turn, and then felt something, and I got scared. So, it went upstream, really far away. Look at that; it went almost back to where it was released, and then eventually tried again, and then escaped. In the constricted, well, let me describe. The fully open experiment showed eels drifting away, and then when they came here, most of them started doing this sort of milling behavior. Well, should I go through? Should I not? Seems a bit dangerous. I don't know. And then decided to go. Okay. We did some sort of analysis of these trajectories, and the results were pretty intuitive. There were longer post-switch distance traveled in the constricted case, and much longer than the fully open, and there were much higher post-switch velocities of the eels. So the eels in the constricted case, the eels swam away really fast, indicating that they got scared for some reason. Okay, they felt the constricted area as a dangerous area. However, this kind of makes sense. This is consistent with the picture. Accelerations scare, eels respond to accelerations. However, it's not clear whether they responded really to acceleration or to something else. If you map, we mapped all the switch points for the fully open and the constricted case, and they all occurred in proximity of the intake. It's a bit more spread the case for the constricted experiment. But again, even if you allow for some sort of flexibility on how you define the behavioral switch points, none of them really was in the areas of high acceleration. All the switch points were kind of upstream. So the eels clearly did not feel these accelerations. If they managed to detect the acceleration field, they use the system, I don't know, I have no idea about. So this opens the questions. Did they respond to acceleration like salmonids do? Or to something else? If they responded to acceleration, how did they detect the acceleration itself? Because they did not came into contact with it. So very weird. Perhaps they responded to noise. You know, clearly it's a much more noisy environment when you constrict the flow. But Adam did some experiments by mimicking the noise of a waterfall, or the noise you would get in the constricted case with some speakers and microphones. He managed to tune it, and doesn't seem to be the driving matrix. Pressure gradient could be another option, or perhaps it was a shortcoming of the experiments. If you go back to the bathymetry map in here, there is sort of a scoured region in here, which is, with respect to background, probably 30-40 centimeters. There is like a slight deepening of the bottom. Perhaps that generated some sort of flow separation or something that would destabilize eels. I don't know. It seems unlikely because it really occurred over large longitudinal scales, so it's not my preferred explanations. Not that I have one, but I think this is unlikely. It's a possibility. Yeah, Gabi was suggesting pressure gradients, but again, from the modeling and the measurements of the flow depth, the gradient in the water surface profile was not that severe in correspondence of the switch points, or if it was, well, we don't have a way to quantify it, so I can't really tell. But it didn't seem definitely there was not a sharp change Okay, pressure gradient. So we checked that, but I'm not sure is the right answer. Also, another colleague of mine is convinced it could be pressure gradient. So we need to dig further on that. The other interesting result, which is my favorite in this study, is that, as I've shown, all the eels liked to swim over these two corridors in the upstream, let's call it in the undisturbed part of the channels. None of them decided to swim at the, in the center. The question are two questions. Why did they choose to swim that close to the banks? And if they were swimming at night conditions, zero visibility. They did not come into contact with the lateral banks. If they decided, so they wanted themselves to swim close to the banks, how on earth they did detect the banks? How did they know that there were banks over there? Because it was completely nighttime and they did not touch the banks. So the current hypothesis that we have is that the proximity to lateral boundaries could be a way as like a navigational cue. So they know where the boundaries are. They know they are swimming in a channel, in a river, and they are following the main current. That's our explanation. But yet, again, the second question is, eels then probably use the mechanosensory system, the lateral line, to detect some sort of hydrodynamic footprint of these, or some sort of a hydrodynamic signature they could detect to feel the lateral banks. And to me, what generates a signal that comes from the lateral banks towards the center, towards where they were swimming, are perhaps the secondary currents. So perhaps they are able to sense secondary currents, or the shear stress that they generate at the bottom. Remember that they like to swim close to the bottom, so perhaps they are able to sense some enhanced friction with respect to the channel center. I really don't know. That was an hypothesis we threw in the table when we were writing the paper. I don't think it's written in the paper. So just to conclude, eels seems to respond to hydrodynamics. There is no doubt on that, but the hydrodynamic metrics they respond to is not clear. Acceleration alone, as for salmonids, doesn't seem to be a candidate. Eels swim close to lateral banks, but how do they detect them? I don't know. What is the hydrodynamic signature they manage to identify? I don't know. And these are really these are open questions, and in my opinion are really, really important. Imagine that we understand. So with these results, we know that they like to swim close to the channel banks. So this is something that attracts the eels. You remember at the beginning we say we need to understand what attracts and repels. So here we have the two ingredients. We know that the constricted case repelled more than the fully open case. Okay, so we have some data showing that, and we know that they really like to swim close to the banks. So if they really like to swim close to the banks, what is the hydrodynamics that attracts them? If I manage to if I manage to reproduce that artificially by putting like stripes of roughness, for example, in proximity of the dam, perhaps I can fool the eels and guide them towards a safe passage, creating artificial secondary currents. And if I understand what really repelled eels from in the barrack, so I really know what is the metrics that generated these rejections, then I can find hydrodynamic ways to implement them in proximity of dams, and therefore prevent eels to go to screens or to be trimmed into the turbines. So I think this whole study really integrates fundamental science with clear engineering applications, so to me it's really, really interesting. And besides the paper that I have already mentioned, our paper, there are also a couple more references to look at that I have used for this presentation. And for me, this is the final slide. I thank you very much for your attention, and as usual, if you have questions, feel free.